This is something we have started a few years ago when we developed methods to electroporate RNA into cells. So we were at that time primarily, and still are, interested in dendritic cells, but we also used that to introduce T cell receptors or CAR T cell receptors into T cells by using RNA transfection. And at that time, people kind of were laughing at us because everybody stated, why should you use RNA transfection? Because it's transient, so the T cell receptor or CAR T cell receptor is present on the T cell only for a limited period of time. When you can refer to or rely on viral transfection. But our argument at that time was already that there might be safety problems, and actually that turned out to be true. So if you put in T cell receptors, it may happen, or even CAR T cell receptors, it may happen that you get toxicity, and that has happened in clinical trials already. And then you have the problem that you don't get rid of the living and replicating virally transfected T cells. So our idea was, if you have many candidate T cell receptors or CAR T cell receptors to screen, let's do that by using RNA transfection. And that was the idea. And later on then we came up with the idea and possibility to put in two T cell receptors, for example, as we have done now, a conventional T cell receptor and a CAR T cell receptor, to test whether this is possible. And it was possible. And we also could put the RNA into gamma delta T cells, which has a big advantage because there there is no danger that there is a mispairing of T cell receptor chains, and so no danger of autoimmunity. So far these are only in vitro studies, so what we saw was that by performing such a double hit, you get much better killing of the tumor cells, right, as expected. And we are planning clinical trials with these transfected T cells. In theory, there are no limits. Of course, what you have to do on the long run, if this in principle turns out to be very useful, that you work on mutating the RNA and modifying it so that you get a stabilized version. So that's possible, and we have already done that in other cases, so that the RNA then stays in the cell for days and even longer. Yeah, right now, with the unmodified RNA, that's for several days. But so for a clinical trial, that's enough. So the transience is, something that's good for testing, because if you are confronted with toxicity, the toxicity will wane, so you don't have any problems. And even if you want to get clinical results, you just give another shot of the T cells, every few days. That's no problem, because you can grow up tons of those autologous T cells and transfect them, so you freeze them down and give just serial shots. So that's certainly not a problem. And you would then, of course, also combine maybe with checkpoint blockade, so you destroy the tumor, etc., etc. So of course we would not go for T cells or CAR T cells that see antigens that seem non-dangerous. So we, of course, would go right now for the so-called MCSB antigen that has not been exploited, and I think people refrain from that because MCSB is also expressed on normal cells to some extent. So at low levels, and probably that doesn't produce any toxicity. But the nice thing is that MCSB is expressed on melanomas, on neuroblastomas, and on the tumor vasculature of virtually all tumors. So if it works, it's really something to go for. I think then also from a production point of view, right? We have a lot of experience in GMP production. You would rather come up with different batches of T cells, right? Because there is a limitation to optimizing things, right? If you put in 8 RNAs, it's the relative quantities, etc. You have to test that out. That's too complicated. So you would deliver and administer different batches, and each batch has 2 receptors. And the idea, to put in 2 receptors is to hit also tumor cell clones that have lost one or the other antigen, right? That's the idea. We are pursuing this adoptive T cell therapy program, and our main program, we have been pursuing for a long time, and we started that together with Ralph Steinman, is the dendritic cell vaccination. And vaccination and cancer vaccines in general have not been so fashionable in the past few years, but now as checkpoint blockade has become available, and it turned out that you don't get success in all cases, all types of tumors, and even if a tumor is responsive not in all patients, it's getting more interesting again. So we are interested in melanoma, cutaneous melanoma, and also uveal melanoma. And uveal melanoma is notoriously resistant to checkpoint blockade antibodies, as far as they are available right now. So you rarely get responses to anti-CTLA-4 antibodies or anti-PD-1 antibodies, or even the combination of those. And so what we have started is adjuvant vaccination. That's a phase 3 trial, actually. And the reasoning for that is there is no therapy available, no effective therapy available to treat metastasis as soon as they have occurred in uveal melanoma. And there is also no therapy yet available to prevent the metastasis, so no adjuvant therapy. And so we have designed a trial where after destruction or removal of the tumor, the primary tumor in the eye, if it classifies at high risk, which is indicated by a loss of 1 chromosome 3, we then prepare a vaccine out of the tumor RNA, which is put into dendritic cells, and people get vaccinated in a randomized fashion. So half of the patients do not receive the vaccine, the other half gets the vaccine.